I hope everybody is connected again. So, I'm Mariangela Russo, and I'm the chairman for this session. We already have an amazing session today, following the amazing session of yesterday. And let's continue with really interesting, great talks. Our next speaker is Manuel Valiente. From the National Cancer Research Institute of Madrid, who's going to speak about making your research translatable, the example of RENACER. So, Manuel, please. Please remember that if you want to ask any question, write in the chat or at the end of the presentation, raise your hands and unmute. Thank you. Please, Manuel.
Thank you, Mariangela. Thanks, the organizer, for the opportunity to present today in this very interesting conference. And I have really enjoyed the format so far, and I hope that I also got a lot of questions, because I think this is a very usually very complicated task of for basic scientists to translate their findings. Usually there are tons of problems, and I hope I will be able to tell you that I was there, and we somehow find our way to create a more efficient way of validating our findings in the lab by using some strategies I would like to discuss today. So I have the disclosure to make, which is not connected to the content of my presentation. So RENACER is about brain metastasis, and I need to give you just a few slides to introduce to you to this field. That these are secondary brain tumors. They are cancers that are very prevalent, that are the main sources of brain metastasis. So these affect many cancer patients. And the problem of this progression of cancer is that there are difficulties to make these patients to survive when they are diagnosed with brain metastasis, even if they have extracranial metastasis, which is the red line here. It really goes even worse when brain mets are diagnosed. Survival is decreased. Treatment for brain mets, as any other treatment for the brain, is really not ideal. There is a lot of cases with resistance to therapies. And also, on top of that, the quality of life of many of these patients is severely impaired, because you have a loss of function on the activity of one of the most important organs in your body. And that creates significant problems, not only for the patients, but also for the families. So it is true, of course, that there is major progress on systemic treatment in oncology, and we can see that the immune checkpoint blockade, for instance, allow patients, for instance, with melanoma to be cured, potentially cured from metastasis, from metastatic disease. But I can tell you that it's more and more frequent that there are relapse to these therapies, and one of the main organs that are affected by relapse is the brain. And that means that therapies are, even more modern therapies are having a limited impact in brain metastasis. And that's why we want to really develop let's say, an effort to personalize therapies to focus on the specific organ where the metastasis is growing, because we think that there are specific vulnerabilities that we need to take into account. So this is the rationale that allow us to think about the RENACER, which is basically a national network to obtain samples from patients from brain metastasis. This network has many hospitals involved. We are oscillating, but now we have 18, 19 hospitals, and these hospitals are sending us samples that they obtain through surgery, and they are shipped all to our institution at CNIO through the biobank. And the particularity of RENACER is that we receive fresh samples. These are not fixed samples. We receive the tissue alive, and we can do things that will not be possible without this strategy. And I will go into the specifics. I will give you examples and so on. We are also collecting liquid biopsies, and just to give you an idea of what we have achieved so far. So we are relatively young in this aspect, but we have collected more than 300 brain metastases. These samples are shipped to us within 24 hours, and we are collecting tissue and blood CSF as liquid biopsies. And with this tissue, we do a lot of things. We are creating a very rich cohort that not only generates FFP samples, but we are also processing all these samples to generate omic data, RNA-seq, collection sequencing, and also we can take advantage of that these samples are fresh, and we can generate what we call patient-derived organotypic cultures, which are crucial to perform not only descriptive experiments, not only descriptive analysis, but also functional analysis. So I was thinking that you might be interested in getting to know RENACER and why this network, this clinical network is needed, and obviously how we went from nothing, from a laboratory to a network with 18, 19 hospitals. I understand that you might be interested on that, but I also will try to make my best because you might be joining this talk because you just want to learn how is the more effective way to translate your findings in the lab to patient samples, or you might be a senior postdoc or a junior PI that is really very interested in establishing a clinical network. And I think I might be able to help you through the presentation and afterwards through the Q&A. So I have designed my talk try to answer this potential interest, and I hope I will manage to do that. So I will give you a few messages throughout the presentation that are covering aspects that I think are crucial to take into account. The first one is really that you might be interested in getting samples, and you might initiate your efforts calling to the doors of many hospitals, and then maybe it becomes a little bit complicated. So of course, my first message is that go to the simplest. There are so many aspects that you need to take into account in order to get high quality samples and get to know the particularities of the clinical, of hospitals that really is a good training period if you just start working with a single hospital before going to many hospitals simultaneously. I think this is something that we have done, and I can clearly see that if we had started in a different way, we had made many more mistakes. Other thing that I think is crucial that you can take into account from the beginning is that you are receiving samples from the hospital, but you should be thinking on how I return this extra effort from the clinicians to them, to the hospital. So how to make yourself valuable in this collaboration. There are many ways in which you can exploit this. It depends, of course, on the needs of the clinicians you are working with, but just a simple thing as giving them yearly updates on your research progress, so they can see how useful are the samples that they otherwise will just throw them to the garbage, how rich that can be for your research and how they are contributing to move something that was already only in the lab to a potential validation in human disease and eventually potential clinical trials. I think this is really, really important. So try to think that they really want to see how your efforts are generating things that could be also valuable for them. Because you have to take into account, it depends on country to country, but they are making an extra effort to help you. What is clear to me is that the most important things to make this type of collaboration successful is really the people. So when we started to launch this collaboration into a national level, we took this screenshot, and these are the, let's say, the founding members of this national network. It's the first time that something like this is done in Spain related to brain metastasis. And I can tell you, and I can see now clearly 3 years later, that if these people would not have been as committed as they are, and they continue to be nowadays, this will not work. So it's really crucial that the first people you start collaborating with, you feel that they are truly interested, that they are collaborative, that they, that you deal well with them, and that you understand each other well. I would like to, for instance, just go back to my initial slide in which I was telling you that start with a single institution. So I started. I was super lucky to collaborate and to find, close by here in Madrid, to start working with Hospital 12 de Octubre, which is one of the biggest in the country, and with clinicians Ángel Pérez, Juan Manuel Sepúlveda, and also Aurelio Laín. These clinicians were looking for potential collaborations, and also at the institutional level, they have somebody who was encouraging clinicians to start collaborating with basic laboratories. And I think this was crucial because they were feeling that they were doing something else. They recognized the problem of brain metastasis. They recognized that the sample where the samples they obtain in surgery, Angel is a neurosurgeon, their samples were just not, or they can give more information than the information they were obtaining from the samples, and we were starting to collaborate. We were 5 years interacting to each other. They were doing the surgeries. We were on our car going to the hospital, taking the samples, going back to the lab, discussing about the sample, attending to the board of the weekly board that they have on the samples that they were sending to us and so on. So it was really a fantastic 5 years to get to, to shape this collaboration, to take care of the things that were not working, to optimize all the things, from the courier to how we were doing the analysis of these samples, right? And then we started to realize that the needs in the laboratory were increasing. More people were joining also, and we realized that we really need an extra amount of samples in order to validate our findings. And we started to interact with other hospitals before launching the national effort. And for instance, we got in contact with Hospital La Princesa in 2020. Now, how to engage other people. I think this is also come from to the beginning of this collaboration. It was the clinicians, it was the surgeon who contacted a friend that was working in another hospital to tell this other surgeon how fantastic was this collaboration, and that allowed the Jose Fernandez Alén to get interested in such collaboration that was already working. So I think that at the beginning is really a little bit of luck, of course, but also to keep this interaction alive, to engage the clinicians, to let them facilitate them to see the benefits of being and doing this collaboration, and also to to make try to understand their needs in order to get to an understanding, mutual understanding that will facilitate for sure the collaboration. Because there are, as you can imagine, during all these years, there have been many samples that were missed until we had the protocol fully optimized. And then 2021, it was the big change when we decided that it was not enough with these 2 big hospitals, but we really need to go bigger than that. Then I was thinking that, brain mets is a huge need. There are no statistics of brain mets. There is no ongoing effort at the national level. So why not, if we need to grow, let's grow, as big as possible and go at the national level. And at that moment, given the amount of work that was working with 2 hospitals, I realized that we needed extra help. And at the moment, I contacted the biobank without not knowing very well whether I was doing the right move. But it was, I can tell you now that contacting the head of the biobank at that moment, Eva Ortega Paino, it was really absolutely crucial. I mean, it would have been impossible to have additional 15 hospitals without the help of a biobank. And this is something that I would like to emphasize. I think sometimes I have the feeling that many of my colleagues, when you mention biobanks, they start to get nervous because they associate that with a lot of paperwork, a lot of difficulties in order to get samples and so on. My experience is completely different. My experience with the biobank is really what has made possible to get, from an initial email of an interest from a hospital to get helping them to go through the paperwork, through the ethical and legal requirements to get approved, to take care of this very complicated shipment of fresh neurosurgeries that and to make sure that every single sample that we receive at CNIO is in perfect conditions because they are temperature controlled and we have very good samples. And then to go through the, to multiply the sample in many tiny pieces and many different sources of information that you need to, catalog in a correct way. So, 10 years from now, we can just go back to the biobank, ask for the sample, and a collaborator in the States, for instance, can also apply and get the sample. So this really would have been absolutely not possible without the help of a biobank. You have to also realize that biobanks nowadays are going to be crucial partners in oncology, in research, in cancer research. They are going to play a very important role. They are paying a lot of efforts in, helping us to work with human samples and also making sure that you are doing this in the right way with all the legal and ethical requirements accomplished. And this is really fantastic to get help from them. And it's beautiful that all these samples, and I will emphasize this later, all these samples are will be made available for the scientific community, and this is because they are in a biobank regime. Okay, these are not my samples, which I think is very, very important. It is true that if you're a PhD postdoc and you have very exciting data that you want to translate, for sure there are different levels of translating those findings to patient-derived material. For instance, you can find in your mouse model that, for instance, in a brain metastasis, these CD8 T cells are just surrounding the metastasis but not getting inside the metastasis, and you find a potential mechanism that is playing a role on that, keeping these T cells far from that, and you can just validate, as you can see in this sample, that this is totally the same in human brain metastases from different sources and so on. No? So this is fantastic. You have validated your mechanism in a descriptive way, but I can, I can tell you that nowadays it's possible to do functional experiments in human-derived material. You can really do the experiment in human samples. For instance, if there is a molecule that is mediating this phenotype and you have tested a molecule, an inhibitor in mouse experiments, you can do the same in human samples. And I will try to give you about this. But obviously, I think that is fair that you get to ask yourself how far you want to go in terms of the validation. And I think in order to decide how far you want to go, I would like to cite or to follow the strategy that Quentin Tarantino has put forward, that he was committed to just direct 10 movies across his entire career. Now, what is the most important thing for Quentin Tarantino is that the quality of each of these movies is the maximum possible. I think this message of, at least myself, being aware that my contributions in science will be limited, and my decision is to make or try to make out of each of those the best possible, to go as far as the story allows me to go in terms of validating our findings to human samples and to even go to clinical trials. Obviously, if it's possible, this is what I want to do, and I want you to think about the quality nowadays is much more important to quantity, especially when you are working on a problem in oncology and a problem such as a brain metastasis, because we just want to find something that helps patients. And if I find just one thing, for me it will be just I will be completely satisfied. I also want to give you an example of some of our efforts on how we are doing this, and in which all the decisions that I just shared with you were involved, and how we do functional experiments in patient-derived material. So, for instance, we were interested in understanding resistance to radiotherapy. That is a cornerstone of the treatment in brain metastases. And we started to collect samples, human samples, through RENACER. to validate a molecule that we identify in experimental models to be potentially a mediator of resistance to radiotherapy. Now, working in such a network, we were able to collect a very specific subset of samples that were tested to be resistant in real life. So patients received radiotherapy and they did not respond. So we collected those samples. And the first thing that we did is, of course, to do the descriptive validation. So we found that this mediator, S189, was highly enriched in those patients that were not responding to radiotherapy. However, what I would like to tell you is that we can, with this type of effort, you can go farther. You can really test whether inhibiting this mechanism could be a novel therapeutic opportunity to improve sensitivity to radiotherapy in patients with brain metastasis, because we are receiving these fresh neurosurgeries. We established these patient-derived organotypic cultures. Basically, these are slices of these brain metastases that we can keep alive for some days in the lab. And there, you can basically identify drugs that are targeting the mechanism of resistance that you have identified and really perform the functional test of whether this drug increased the sensitivity of these radio-resistant human brain metastases, proven to be that in real life, to see whether you can sensitize them for this combination. And you can find this, fantastic data by which taking these 7 patients that proven to be radioresistant in real life, just the radiotherapy applied to these organotypic cultures doesn't do anything. So the squares are white. The drug doesn't do anything by itself, but if you test the combo, then you start seeing that the experiment, the data you generated in experimental models can have really the possibility to be translated to patient-derived, to patients. This is really to go one step further, and this really facilitates the possibility to run clinical trials. And I can tell you that this has been a very fantastic experience in how just publishing the paper very shortly after that, we find right partners, both clinically, but also at the pharma level, that were inspired by our data to start a clinical trial, and this is ongoing. And I can tell you that the clinicians that are involved in this trial are clinicians that are part of the network. So they knew the story from day one, and they know all the data because I have been, giving them updates and so on. So they are committed from the very beginning. I think this is really increasing the efficacy of translating our efforts. You can also think about biomarkers to be exploited. Is equally important, as Mauricio was saying before in the previous talk, to find biomarkers. In this same project, we found out that we were able to, by looking at liquid biopsy, measure the levels of this biomarker and predict in retrospective samples which patient is going to be responding poorer to radiotherapy according to the levels of S189. This is a retrospective study. So if you don't do anything else, pharma companies or clinicians will not be particularly interested on that. So you really need to do the prospective study. And I can tell you that it has been very, straightforward to encourage members of the network to launch the prospective study of collecting samples from patients. So this is non-invasive study, collecting samples from patients that are enrolled in the standard of care. They are diagnosed with brain mets. They are going to be receiving radiotherapy. So we collect the samples and we can validate our retrospective study in a prospective study. And this is ongoing. We have collected more than 70 patients, and this is ongoing. And really, we can make a big impact being an academic lab, because we want to find out if this is proven correctly, the first biomarker that could predict resistance to radiotherapy from a non-invasive sample, from a liquid biopsy. And I think this is really, really inspiring. So for those postdocs or those junior PIs that are thinking about, developing a similar strategy, obviously you need to know that there are many things involved into the expansion to the national level. We get to know each other. We do general assemblies. This is just a part of the people involved in RENACER. I'm sure that we are reaching probably 200 people involved And I was thinking about the things that this involves, and I can tell you that there are many things that you have to do that you were not doing before establishing such network. All of those are, really fantastic things to do. I just want to to emphasize a few things that I think is hopefully motivating some of the people in the audience. I think this allows to think big. It was mentioned that only in pharma you can do this, but I don't think it's the case. You can generate a real impact. I think you can do this being a basic lab, as I consider myself. I'm a neuroscientist and a cancer researcher. Something that is really also very enriching is that you really need to be inclusive. You need to deal with many people, from nurses to people working, transporting your sample, pathologists, mathematicians, statisticians. So you really need to understand the needs of many people, and this is just making yourself more to have a different vision of things. And I think this is in science. It's really fantastic. It's a privilege. I think that this also opens things that maybe you were not thinking at the first day, of course. For instance, I told you about our efforts to generate to do omics on these samples, to collect all these liquid biopsies. But for instance, now we are expanding to collect all the images of the patients that are, diagnosed with brain metastases, and we can even now collect the cognitive status to create a database which is not existing now. It's a database that will register the status, the cognitive status of patients. And what is more important is that for the first time, we will be able to do the integration of all this data, from omics to imaging to cognitive status, liquid biopsies, tissue samples, and so on. And this is very unique of RENACER. And I think the more important thing is that we are doing this to facilitate the access to clinical information. I think nowadays this is going to be the rule for anybody who want to establish such a network. We are doing this thinking that we are going to create a data portal. All this information will be available to the scientific community, the omics data as well. People can apply to collect samples, to request samples. We are making a big effort also to communicate what we are doing in the network. We are creating multiple channels, and we have also a web page that was launched very recently. And I think this is really something that we all need to do. No, I don't think that somebody who is collecting samples will have the possibility nowadays to do what has been done in the past, to keep the samples for their own purpose, because those samples are not belonging in the first instance to the PIs, belonging to the patients ultimately. And I think getting immersed in these efforts, really seeing that other laboratories could benefit from that, to increase the knowledge and to improve the research in brain metastasis, I think is the most important thing that you can get involved with. Also, of course, if there are people thinking about this, to apply this to other metastases, we are seeing this now in brain metastasis, where we see other efforts that are ongoing in different parts of the world. And why not thinking that we can connect at some point efforts that are taking place in other parts in Europe with efforts that are taking part in the States, try to connect, try to share samples, try to increase the amount of data to get our conclusions even more relevant and more representative for the patients. I think that this can go even to metastases outside the brain, and it will be really fantastic that we can create these interconnected networks. So I started the presentation trying to identify the needs of the audience that might be seeing this presentation. I think that the most important thing that I want to mention is that I recognize that it might be difficult to translate your research to patient data, but please try. I think that it will be really satisfying to see whether your molecule is validating in human samples, and even now that you can do functional experiments, and then maybe if you do that, this can move into a clinical trial. I think you really need to try. I think you have to get the highest quality possible if your project, of course, allows to do that. You need to convince your PI, you need to convince your supervisor, try to do that as well. I think there are ways to do this, take advantage of the biobanks. And yeah, I think I will finish here. Just want to thank the many people involved in RENACER, many different people. Of course, the most important people are patients here that are donating their samples in a very complicated moment. The funding agencies that are allowing RENACER to keep growing. And thank you very much for your attention. I will be really happy to engage in conversations with you.
Thank you so much, Manuel. This is really inspiring presentation and really a great example of collaborative work and huge effort. So please, if there are any questions, you can type in or you can simply raise your hands. While we wait for questions from the audience, I have a couple of questions to break the ice, I would say. So my first question is, obviously, this is a huge effort. There are multiple centers that contribute to this huge biobank of samples. So what's the policy related to samples for which there's limited material? How do you decide the priority of the type of assay, for example, on paraffin embedded samples, for example, immunohistochemistry that you need to perform? How do you prioritize if there are many requests for specific type of samples and there's limited amount of material? How does it work?
Yeah, so as I said from the beginning, we think that the cohort that we are creating should go beyond the lab, and our main interest is to generate the most useful cohort of brain metastasis samples. So we always prioritize those that are going to last forever. Let's say the processing for omic, like RNA-seq and whole exome sequencing. We have already profiled, as I said, more than 300 human brain metastases. We have the clinical information, and we are going to create with that a data portal. And also we create we prioritize also the paraffin embed samples, because this can be useful for many. I mean, this is the most frequently used sample nowadays, and many people might be interested to get that. So there are always priorities, and we have that very well established, and this is the criteria.
Okay, thank you so much. We have a question from the audience. Anuya, would you like eventually to turn on your camera and ask your question by yourself? You have been allowed it to. Okay, okay, I will read it. So thank you for a very interesting talk. Probably she's here. Anuya, she or he, sorry. Would you like to ask the question by yourself?
I think I'm not the right person.
No, there's also Anuya. Okay, let's go first for Anuya question, and then I will give you the chance, George. So thank you for a very interesting talk. I have a question rather regarding your inhibitor study. If I understood correctly, patients with relapse were non-responders, and combination therapy with radiation made them sensitive to the treatment. Does this mean your inhibitor would alone be enough for the primary metastatic tumor? Was this tested? And then second question, does the sensitivity for the combination therapy varies based on the origin of the tumor tissue?
So the second one is no. So we have data which is not dependent on the primary source of the metastasis. And I want to also use this question because I think that there is a real need in the possibility to personalized treatments, but focusing on the metastatic disease, not much on the primary source of the metastasis. We have very encouraging data in which we are able to see that vulnerabilities of brain mets can be completely dissociated from the primary tumor. And I think this is important efforts to do. Regarding to the first question of whether the drug, the combination with radiotherapy was already effective in the first, without the going to resistant samples, I mean, we only tested resistant brain metastases because we have data suggesting that the mechanism could be facilitated by just the radiotherapy itself. We see significant upregulation of the receptor that is inhibited by the inhibitor when you give radiotherapy. And so this is what we tested, but it remains to be seen whether combining this from the very beginning might be effective. This is something that we are doing now in this trial as well.
Thank you. And that's George. Can you please turn on your camera and unmute? Ask yourself.
Yes, I cannot use my camera right now, but
that's fine. That's fine.
So yeah, I have a couple of questions myself. So you mentioned, first of all, thank you very much for this talk. It was from every point of view, it was quite interesting. You mentioned at some point at the beginning that the transfer time from the hospital, of the tissue from the hospital to the laboratory is, did I get it right? Up to 24 hours, I think. And it's alive, the tissue is alive. That was quite remarkable, actually. But I was wondering, during this time, if the tissue is alive, can't you possibly lose some important information? For example, if the tissue is alive, then mRNA will continue being transcribed, and there's going to be turnover. Metabolites will continue changing in there. So in what state are the tissues transferred to the lab?
For sure, it will not be exactly the same as if you process the sample directly from the operating room, that's for sure. But in order to allow these 24 hours, we did some initial tests, because as I mentioned before, when we were collaborating with a single hospital, we were going to the hospitals after a phone call, we were going there, and it was faster, no? And so obviously we started to test in a few selected samples, whether the sample immediately processed compared to the same sample stored 24 hours, whether there were differences, not only at the histological level, but also we profiled those samples with RNA-seq. And the bioinformaticians were unable to differentiate between the 24-hour sample or the 3-hour sample. And I can tell you that now we are thinking on the possibility to expand this beyond the country. So we are thinking that it could be a fantastic possibility if we get to obtain fresh samples after a surgery in a patient with brain met that is enrolled in a clinical trial, and we test the fresh surgery in the laboratory for the same drug that is going to be given in the patient to control, the potential relapse. And of course, that involves that since this is transport between different countries, we have increased the timing to 96 hours. So we are using a special media that is published. You can find this media in our publications, and there are other laboratories that have used this media. It's enriched in multiple factors and so on. But the tests that we have performed, ensure that we are processing the samples with good quality, and that we don't see differences between these 0 or 24 hours.
That is really remarkable. Thank you very much. If there are no other questions, I could follow up with mine. I don't know.
Yes, please go ahead, George.
So this is quite interesting that you obtain these metastases from the patients. But I was wondering, do you also recover the pieces from the primary tumor site, and you make comparisons between them?
Yeah, so this is, for instance, one limitation of what we are doing. So obviously the type of patients that we are studying are patients that have a very complicated clinical history, meaning that, for instance, a breast cancer patient that have a brain metastasis might have had the primary tumor diagnosed even 10 to 15 years ago. And in order to do this in a proper way, it's really, really complicated. Even though, a lung cancer patient that is diagnosed initially with a primary tumor and then with a brain metastasis, even though that this is close the diagnosis of brain met is close to the diagnosis of brain met tumor, usually it involves different departments in the hospital, and coordinating that it has a very significant increased number of things that you need to manage well in order to make them work. So unfortunately, it's something that we are not doing, but it will be really fantastic. Yeah.
And one last question when it comes to the organization of this entire network. So you recover all these tissues, you make this tissue bank, which I'm sure it takes a lot of effort and energy for each individual lab to store them, to manage them, to make the right inventory and all that. So is it possible to centralize this entire process, make it probably state-funded, and more organized, because each researcher has a billion things to do in a day, and as the network grows, of course, this requires even more of their time. So how possible is this in a European context to make this more centralized?
Yeah, thanks. This is a very important question. So there is a moment in which this will need to be, as you said, funded at a different level to make this a stable effort that will remain, over the years. Otherwise, it will not be possible. So this is our goal. So we are trying to make this to consolidate this effort to to publish the benefits of having such network, to create this data portal, to make this discoverable for researchers, and obviously then our strategy will go to the national health system to see whether this could be something that they can assume. Because as you said, initially, okay, we were 100% involved in this. Now we are lucky enough to have a biobank, which is taking care of, an amazing amount of work. But if you want to keep this going, it's absolutely necessary that this is in place. And I can tell you that getting funding for this type of effort, it sounds less exciting as an effort to, treat, or to develop an experimental model or to target a molecule that you have found. So the current funding schemes is not ideal to fund this network. So we were lucky that Asociación Española contra el Cáncer and La Caixa allow us to get funding specifically for this, but we had to get to a site in which that was evident, that was worth funding. But it's challenging. It's very challenging.
I understand. Thank you very much. Thank you.
Thank you.
Thank you. We have one last question from the chat. Thank you for your talk. My question is related to the combined chemoradiotherapy. Did you also look into the mechanism of the enhanced combined effect? Did the inhibitor enhance the radiosensitivity, or did the radiation treatment enhance the effect of the inhibitor?
So what we see, I don't think I can answer this question in detail. So what we have seen is that basically these cancer cells, when they are in the microenvironment, they are getting help from the microenvironment to activate and produces 189. That signal through RAGE receptor and activates inside the cell an NF-kappa beta dependent mechanism that involves JUMB and that facilitates DNA repair. So what we are doing with this inhibitor is basically to block the action of S189 through RAGE and blocking the increase in NF-kappa beta signaling pathway. This is as far as we have gone. There is still need to dissect very in detail the molecular mechanism, but for sure we are blocking a signaling pathway that is highly involved in resistance to radiotherapy, but to many other therapies as well, which is NF-kappa beta.
Thank you so much. And
I have a question.
Yeah, okay, very quickly. So we need to move to the next speaker, but please go.
Okay, thank you for your presentation. And as someone who has seen a lot of tumor tissues as you. What do you think about the ability of organoids for mimic these tumor tissues?
Well, I think, thank you for the question. I think organoids is a fantastic tool. You can do things that maybe we cannot do with our tissue slices. For instance, our tissue slices are short-lived, and we cannot just give them in the fridge for doing more experiments. However, our patient-derived organotypic cultures is fully compatible with clinical decision making, meaning that if I receive a sample, I process the sample within the same day, I use an inhibitor, and I know whether the patient-derived material is showing me a response to treatment, and I can eventually notify the clinician that this drug is giving an effect. We have found in some previous publication that the data you collect in patient-derived organotypic cultures related to sensitivity to drugs is predictive of what is going to happen in the patient. We have done this study in glioblastoma patients, so we were able to predict whether a glioblastoma patient was going to have a better or worse response to temozolomide and radiotherapy, because we gave the same combination therapy in the patient-derived organotypic cultures. And we were able to predict responses or non-responses in almost 9 out of 10 patients. So these organotypic cultures has a lot of potential. Of course, this study is a small study. We had like 18 patients, but we are now expanding this to get into clinical trials, real clinical trials, to see whether we can predict as quick as possible whether the patient that is enrolled in a clinical trial is going to respond to the experimental drug or not. And we can notify that to the clinical trial, to the clinician, to see whether we can offer other possibilities to the patient eventually.
Thank you for your question. I have one more question, but we don't have time, I guess, so I email it.
Okay. Yeah, I mean, you're absolutely free to email Professor Valiente. Thank you so much. So thank you again. This was the great result of an amazing effort. So thank you so much for sharing it with us.